our previous co-organizer, Kevin Yang, joining us to kick off the season. He joins us from Microsoft Research New England, where he works on protein modeling, made a lot of contributions to that field, broadly speaking. And today he's going to be talking about one of the new preprints out from his group called Protein Sequence Generation with Evolutionary Diffusion, EvoDiff. So Kevin, without further ado, take it away.
Thanks so much, Chase. And it's great to be back at the seminar. I say back, but this is actually my first time presenting. I'm used to hosting and running the Zoom and letting people into the room and all that. So today I'm going to be talking about protein sequence generation with evolutionary diffusion. And I'm sure everybody here would agree. We, as a society and as scientists and as engineers, we want to design proteins that do new and useful things. Some examples of things people have done or might want to do are things like new chemistry. The example I'm showing here is using a heme protein to make carbon silicon bonds, which are not known in nature but are useful. And this is out of the Arnold lab. And we want to do this in the future, not just new reactions, but also we want to make our reactions fast and selective. And we want to do this mostly for environmental reasons. We want to use enzymes instead of heavy metal catalysts. Maybe there are reactions that would be really nice to do, like breaking down all sorts of plastics very quickly, or things like that. I'm going the wrong way. We also want to use proteins as therapeutics. For example, this is asparaginase, which is an enzyme that breaks down the amino acid asparagine. And the reason that works as a therapeutic is that many cancer cells can't make their own asparagine. So if you have something that breaks down the asparagine, it kills the cancer cells faster than it kills the healthy cells. So this is a chemotherapy drug. We also use proteins as molecular tools, the most famous of which is green fluorescent protein, which is first discovered in a jellyfish. And this is useful because you can bond it to things, tag it to things, and then you can visualize where things are under a microscope. All right, so one way we can use machine learning to generate new proteins is to take what are called diffusion models and generate structures. So what this works, how this works is in a diffusion model, you start with some item. We're going to call it X0, which is your original thing during training. It's a real thing. And this might be a structure. It might be a structure represented as an angle, as a set of angles, or as a bunch of coordinates, or frames and rotations. And then you have a forward process, a corruption process, that step by step adds noise to this real thing until it's indistinguishable from a sample from a unit Gaussian, in this case. And so we can call that just a random structure. And so after big T steps, what you have is indistinguishable from random points. Then you train a neural network such that it sees a partially noised item and it figures out how to denoise it one step. And you show lots of examples of how to denoise things one step. Then you give it examples of random noise and have it denoise it all the way back to something that looks hopefully realistic. And so here we see a structure diffusion model starting from a random set of angles, which is a random structure, and denoising that to generate a new structure. And these models have been pretty successful. They generate very realistic looking structures. And people have shown that you can design sequences of a fold of these structures, and you can make them in the lab, and they actually fold the structures you think they will. And the intuition for why this might be helpful is that because protein function is mediated by structure, if you can design new structures and then find sequences that fold them, you can get proteins that do new functions. The disadvantage here, though, is that structure, while in many cases it mediates function, sequence is really the universal protein design space. And so if you generate a structure, you have to do another step at some point to get a sequence that you can actually go and synthesize in the lab, right? I can't synthesize that structure directly in the lab. I have to go figure out a sequence of amino acids and then figure out the codons I want to use and put that into whatever expression system I want. So at the end of the day, you're always synthesizing an amino acid sequence, not a three-dimensional structure. And when it comes to engineering, oftentimes you want to use a function predictor. So there's a lot of sequence function predictors now that operate in sequence space. There's a whole cottage industry of people doing pre-trained sequence models and then fine-tuning them into whatever function they want. And it's much easier to leverage those if you're working directly in function space. Another reason we might want to work in sequence space is that there's many more sequences and structures. So here I'm showing each dot represents a bunch of sequences. I'm showing about 1 billion sequences, which is probably much less than what we know now about natural sequences, and definitely much less than the total number of possible sequences. If you want high quality deduplicated sequences, you're looking at about 50 million sequences from UNIRF 50. And then, but if you want crystal structures, if you want solved structures, you're looking at only about 200,000 PDB structures. So that's a several orders of magnitude difference. And to make things worse, the structures are not a random sample of the sequences. So those are biased towards things that people have cared about because they're implicating disease, or because they were just things that were easier to crystallize, or because they're from a model organism. There's also the fact that not every function is mediated through a static structure. So many things, such as catalysis, are actually mediated through several conformations or an ensemble of structures. And the kind of extreme range of this are disordered regions, which do a lot of important cellular functions, and they don't really have a stable structure necessarily. So they shift over a conformation or an ensemble of structures. And that sloppiness, that disorder is integral to their function. So you can't design that away and maintain function. So for all these reasons, we'd really like to do design and generation directly in sequence space. And so, as I said, sequence determines the structure, which determines the function. There's been a lot of work recently using diffusion models to generate that structure step, hoping that this will lead to new functions. What we'd like to do is integrate it out, the nuisance variable, and go directly from sequence to function. And generate the sequences and hope you get new function. And this is joint work with a lot of people at Microsoft. So from left to right, Sarah Alamdar is a data scientist with us. So she's the first author in this. She did probably 75–80% of the actual experiments. This project began as an internship project with Nitya Thakkar, who was the second one there. She's with an intern 2 summers ago. She's now just starting her PhD at Stanford. And then next is Alex Liu, Nicola Fuzzi, Ram Vandenberg, and Ava Amini, who are other researchers at Microsoft who helped with this project. So we call our model EvoDiff, and there's 2 parts. There's evolutionary-scale, and diffusion. And so I'll explain each of those parts. So first, protein sequences are discrete series of tokens, right? At every position, you have some choice of amino acids. So it's not continuous the way structure is. So we're going to use a discrete diffusion method instead of the continuous diffusion I showed earlier. The idea is still the same, but we tried a couple different methods actually. So the first is OADM, or order agnostic diffusion, an order agnostic diffusion model. And what happens here is at every step, we mask one position of the protein in the FOR process, the corruption process. Then we train a model, and when we train a model is we take some sequence, we randomly mask some number of positions, and then we predict those masked positions. And then at test time, at generation time, you start from a sequence that's all masks and decode one at a time in a random order. And the key is that during training, it sees every sequence with a different random mask every time that's sampled. So in the expectation, you should have, the model should learn how to do any ordering and should decode any order. Another method we tried is called discrete denoising diffusion probabilistic models, or D3PM. And here, instead of just changing exactly one amino acid at every step to a special mass token, what you do is at every step, you randomly mutate every position. And obviously you can't mutate everything at every time step, but you can definitely mutate more than one thing at every time step, or only one or none. And the probability of mutation at every time step is determined by what's called a transition matrix, which just gives you a probability of going from amino acid A to amino acid B. And once again, your neural network denoiser is going to look at a specific time step and it doesn't actually get told which positions are mutated. And it has to try to reconstruct the original sequence. And by doing that, at generation time, you can start from a random sequence and step by step edit it towards something that is hopefully more realistic. So we tried both of these models and we trained them on this with the same architecture on the same data set, and we do some preliminary tests, and we find that the order agnostic diffusion model does better than D3PM. And we suspect the reason here is that this is an easier corruption to learn, because you're always editing exactly one token at every step, so the model doesn't have to figure out how many positions to change at every step. And it only needs to change each position exactly one time. Once it's decoded, it's fixed. So we call this model EvoDiff sequence. And I've just explained the diffusion part, and the evolutionary scale part is that we trained this on evolutionary sequence space. We trained this on Neura 50, which consists of about 50 million sequences. Those are sampled from many different organisms across the tree of life. They do many different functions. And this is a 640 million parameter CNN, a convolutional neural network. And once it's trained, you can take it, start from a sequence of all masks, and just randomly generate a sequence. And many times that sequence will, if you could run it through a structure predictor, will actually look like what you'd expect a real protein to look like. All right, so obviously we are not the first people to try to train a model to generate protein sequences on a big data set of protein sequences. So next I want to talk a little bit about how EvoDiff-seq is different from those. So EvoDiff-seq generalizes masked and autoregressive language models. And I'm going to explain what I mean here. So in this OADM framework, this order agnostic diffusion model framework, your neural network learns to generate proteins in any order, right? So it should be order agnostic. And it learns every step of that generation process. So one common other kind of big kind of protein sequence model is what's called a left to right autoregressive language model, which I'm going to use LRAR, left to right autoregressive. And the way these work is you start with the start token, and you always generate the next token. So you're always doing next token generation. We can see this is very similar to OADM if you only consider one decoding order, the left to right one. And it turns out that this is a slightly easier task to do because the model has to learn fewer things. It always goes left to right. It doesn't have to worry about the order agnostic part. And you'll see later on that if you just consider unconditional generation, you can get slightly better quality from an autoregressive model. But what going from LRAR to this more general OADM framework does is that let's do conditional generation. And what I mean by that is you can specify arbitrarily which amino acids you'd like to fix, and then generate everything else. Another family of big neural net protein sequence models are what's called masked language models. And these include things like CARP, which shares the same CNN architecture with EvoDiff and ESM, which is the backbone behind ESM fold. And the way these work is at every step, you sample some subset, a fixed percentage of positions to mask or mutate, and then you reconstruct the original sequence. So this is equivalent to learning one step, or in practice, if you sample the number a few steps in the OADM framework. And so when we go from this masked language model framework to the order agnostic diffusion model framework, what we gain is better generation from scratch because I've trained my model to do generation from scratch. And we can look at this if we look at the test perplexity of these models and plotted by the percentage of the sequence masked. So we can see that, so lower perplexity means the models put more probability mass on the correct amino acid in the test set. So at low mass position, so towards the end of generation, OADM and the masked language model do about the same. But at the beginning of generation, when almost everything is masked, because your masked language model isn't trained to do that task, it's not nearly as good there. And in fact, the perplexity is often worse than random. So by generalizing autoregressive language models and masked language models, what EvoDiff-seq can do is we can combine conditional generation with high quality generations. And so we're going to make the claim that EvoDiff enables controllable generation of plausible diverse sequences. And we do this using two things. We use order agnostic diffusion, and we use evolutionary-scale training. And the first thing we're going to do, we're going to evaluate and go through the rest of the talk. We're going to look at how plausible are the sequences we generate, how diverse or how well do they cover the natural distribution of sequences. And then we're going to look at, can we do conditional generation? So first, EvoDiff-seq generates highly plausible proteins. And how we're going to evaluate this, we're going to take our model, we're going to generate a sequence, we're going to run it through a structure prediction module, in this case, OmegaFold. And we can look at the predicted structure, we can look at it, we can also see how confident OmegaFold is. So PLDDT is its predicted confidence. High means it thinks this looks like it thinks it's seen before. Low means it does not. And then we run it through inverse folding, in this case, ESMIF, to get back a different sequence that we can compare to the generated sequence for self-consistency. So we want so here I'm going to plot the self-consistency perplexity against the sequence PLDDT. So we want a high PLDDT and a low perplexity. So that'll be that lower right-hand corner. First, I'm plotting the distribution of test sequences. So you can see there's a pretty wide distribution, but many of them are in the lower right-hand corner, which you would expect because these are natural sequences. And here are the EvoDiff ones. So there's definitely fewer in that lower right-hand corner, but there are still a fair number down there. And if you cherry pick examples, you can definitely find things at various lengths of generation with high OmegaFold prediction confidence and very low perplexities. And then the Hammond distance shows how different it is from the closest natural sequence. So these are not identical to natural sequences. And we'll make the case here that these results, that the exact training we do results in having better sequences. So we have generative training, diffusion training on a evolutionary-scale data set. So here we're going to compare on the left is what you saw before. And here is that left to right language model. You can see that actually also looks pretty good. It might even look a little bit better. There's maybe a little bit more in that lower right-hand corner. So these models have a generative training task. They know they're trained to generate from scratch. And we see that there's a mismatch in the tasks, as with a masked language model, you generate much less plausible sequences. All right, so we've talked about plausibility. So now let's talk about diversity, or do we cover the breadth of natural sequences? So EvoDiff recapitulates the natural functional distribution. So how are we going to test this? First, I take a bunch of test sequences, or these are held out from the training for NeuroF50. I run them through PROT5, which is a orthogonal pre-trained protein masked language model, and I get embeddings for this. And the reason we're using PROT5 is because there's some work out there showing that if you take embeddings from PROT5 and cluster things by nearest neighbor in that embedding space, you can use that to impute functional annotations, Go annotations, to proteins that haven't been seen before, based on proteins that people have already annotated. So we know that this embedding space captures some notion of structure and some notion of function. And then we can, if we want to visualize these embeddings, we can plot a map of them on the right. Okay, and then, so these are the natural embeddings. Ideally, if I do the same thing from generated sequences, I'd like to kind of cover that distribution and show that we're pretty similar. And we can evaluate that quantitatively with what we're calling a Frechet PROT5 distance. This is analogous to Frechet inception distance, and it looks at how similar those distributions of embeddings are. Okay, so I've shown one number. That number by itself doesn't really tell you much. So let's compare to some other models. And here we're going to see that having evolutionary scale and diffusion improves the FBD, which shows that we're covering the natural distribution better. So first, the toughest baseline, as always, is that left to right language model. So this one actually does slightly better. But remember that we're going to do conditional generation later, which the left to right model can't do. If you look at the masked language model, you see that the FPD is much higher. And if you look visually at the embeddings, they are also not covering much of the space. And remember that, as we expect, we get much better quality generations from either a diffusion model or an autoregressive model than from a protein masked language model. We also want to look at structure-based diffusion here. And even, so this is RF diffusion is a very good structure generator. You can take structures from there, fold them using, or generate sequences for them using protein and PNN or USMIF, and then plot them the same way. You see that these also don't cover the natural distribution as well. And we kind of expect this because, as I said earlier, the sample, the subset of sequences where we know the structure is a biased sample of natural sequence space. And there's just many fewer of them. So we see here that both the evolutionary scale and the diffusion help here. We can also see this by looking at the predicted secondary structure distribution. So here we can push any set of sequences through ProTrans and have it predict the secondary structure, which is strand, helix, or loop, strand, helix, or other at every position. Here I'm plotting it for the validation set and compare that to the same distribution for the test sequences with a KL divergence on that distribution. So for validation, our KL is about 1.37 times 10⁻⁵. So lower is better here. You want to be more similar to the test distribution. EvoDiff is about 1.5 times 10⁻⁴. You can see again that autoregressive model does pretty well. And then the masked language model, ESM2, and RF diffusion, the structure-based model, do not do nearly as well at matching the natural distribution here. All right, so next, we've talked quickly about, are the individual sequences plausible? Then we've talked about, are, as a distribution of sequences we generate, are they as diverse? Do they capture the natural distribution of sequence, function, and structure? And now we're going to look at several different ways of doing conditional generation. Okay, the first way we're going to do this is we're actually going to train a whole different model that generates MSAs. So this was actually Nithya's original internship project, and then we extended it to sequences later. And a little bit of background on MSAs just stands for multiple sequence alignment. You take some sequence you're interested in, we call it query. You look at a database, do homology search, find sequences that are similar, have parts that are similar, and then you align them. And the motivation here is that these are related sequences. Looking at the distribution of substitutions at every position will give you information about evolution, will give you information about structure. And we know it gives you information about structure because AlphaFold uses these multiple sequence alignments as an input, and you can do much better with MSAs than with single sequences for predicting structure. For the sake of computational tractability, we're going to subsample these to 64 sequences. And then we're going to train a diffusion model. So it's the same idea, except now, instead of starting with just a single sequence, we start with a multiple sequence alignment. But at every step, we just mask exactly one token. And then at generation time, you can do unconditional generation of the entire MSA, or we can do conditional generation, which I'll show on the next slide soon. And we call this model EvoDiff MSA. It's trained on 100,000 approximately open-fold MSAs. Thank you, Mohammed Al-Qureshi, for releasing those. And once again, these are for many organisms, and they cover many functions. And we train a 100 million parameter MSA transformer model here, which is a 2D transformer with tight attention. And the advantage of using an MSA, obviously, is that you get that homology information. The disadvantage is that this MSA data set was generated for the purpose of structure prediction, so there is a bias towards structure domains. And the first obvious application of EvoDiff MSA is to use it as an amortized local generative model. What I mean by that is there are these local generative models where you can take a natural protein family, you train a model, and then you can generate hopefully new functional members of that family. And people have done this with GANs, people have done this with VAEs, people have done this with other regressive models. I'm sure I'm missing some. Pre-deep learning, you could do this with a HMM. You could do this with a POTS model, which looks at the pairwise correlations. But there you have to train a new model for every family. And we're calling this amortized because we train those once over many families. And hopefully it applies to any family. You show it later. We call this evolution guided generation. You take an existing family, you mask out the query sequence, and you regenerate it using EvoDiff MSA. So how do we do this? We're going to show that this evolutionary diffusion here improves this kind of generation over using ESM MSA, which is a masked language model with the same architecture and a much larger training set, or using our other baseline, what's a POTS model. So we take that original MSA, we mask the query, and we push it through EvoDiff MSA to regenerate the query. Then we can run that through OmegaFold to predict a structure. And we can again run that through ESMIF or protein PNN to reproduce sequence and compare how consistent those sequences are. So here's the same style plot. As you go to the lower right, that's increasing plausibility. Here are the validation query sequences. These look more structured than the single sequences because this training set is biased towards structured domains. And here are the EvoDiff MSA ones. So not as good as the natural ones, but still not bad. And you can I'll show you some cherry-picked ones soon. We can also compare. So here's the same data, just plotted one axis at a time, so that I can compare to other models. You can see the bar is the median. You can see once again the validation set is a hard part to meet, but we get a wide variety of PLDTs and perplexities, and some of them are consistent with the natural sequences. So the next model we consider is an ESM-MSA, which is the same model, but now it's a masked language model, so it's not trained to do generation from scratch. And here we're not actually disadvantaging the masked language model too much because we're not generating from scratch. We're generating just that first row, but it still does worse than EvoDiff MSA on both metrics. And we also consider a POTS model, which does actually reasonably well. And once again, the POTS model is a non-neural network model. You take an MSA, you compute, you estimate position-wise and pairwise energies, which allow you to compute the likelihood for any sequence coming from that MSA, and you can use that to generate new sequences. The point here is that the diffusion and just to show that using EvoDiff does give you gains over the baselines here. All right, in addition to looking at those, we can also predict the structure for the original query and see how similar those structures are. So here, TM score gives you structural similarity. And so more similar to the right. So you can see EvoDiff MSA. Sorry, this is an empirical CDF. So the Y value is what percentage of, or what fraction of your data set has at least that TM score. And you can see that compared to ESM-MSA and to the POTS model, we generate structures that are much more similar to the native structure. We can also compare the original query sequence and the generated query sequence. And here we see that in general, our sequences are much less similar. They're about the same similarity as the ESM-MSA ones, but they're much less similar than the POTS model as well, which is interesting that we get more structural similarity, but much less sequence similarity. All right, so, and then here are some cherry-picked examples of these local generations. So the top row is a we take a generated sequence and predict structures, just so we can visualize them. We can look at the original query structure below, and there's some metrics such as the prediction confidence to self-consistency perplexity, the TM score between those two structures, and then the percent similarity between the sequences. So you can see that there are some things we find here with less than 2% similarity, but a 0.94 TM score. The caveat, of course, is that these are all predicted structures. All right, next, we're going to generate intrinsically disordered regions. So as I mentioned earlier, intrinsically disordered regions perform many important cellular functions. Here's that pretty animation again. And when we looked at our unconditional generations, we predicted secondary structures, which I'll bring back here. And you can see that if you use a structure-based model, you really miss out on this part. These residues that are not predicted to be either a helix or a strand. So they're loops or they're something else. But anything disordered would go there. I have to be careful here because just because it's not a helix or a strand doesn't mean it is disordered. So this led us to wonder whether we could purposely generate disordered regions using EvoDiff. So the way we do that is, if we want to use EvoDiff-seq, we take an IDR and its surrounding structured regions, we mask out the IDR, push it through, and just regenerate the disordered part. If we want to use EvoDiff MSA, we have to get an alignment of IDRs, which is actually pretty tricky because they evolve so quickly. Homology search doesn't work that well. Fortunately, one of our authors, Alex, has previously came up with this really clever way to find homologs for IDRs. The way he does that is, instead of trying to align the disordered regions, you go look at the surrounding structured regions and align those, and just assume that any disordered regions between aligned structured regions are also homologous. So we take those and feed them and mask out that the query sequence of the IDR, the IDR of the query sequence, other way around, and push that through EvoDiff MSA to regenerate the disordered region of the query. All right, so we can look at these. We can then take that generated IDR and the surrounding structure regions and push it through a disorder predictor. In this case, we use Dr. Burt because it was fast and not reliant on sequence alignments, and hopefully somewhat reliable, and get for every position the probability of being disordered. So you can see here, we've highlighted the part we regenerated, the IDR in green, and that part has high disorder prediction. The structured part has low disorder prediction. That's as you'd expect. We can also do this for the original. And we show here that, and then you can compare them. In this case, we actually make it more stereotypically disordered than it was before. So here's another example of a generation from EvoDiff-seq. So this is a serine or threonine protein kinase. And you can see the generated one is on top. You can see the sequence, you can see the predictions. The true one is in the bottom. We're only showing the IDR sequence, but there's also a surrounding structure region, which is kept constant. These sequences are only 17.7% the same, but we managed to preserve the disorder predictions in the disordered region and the order predictions in the ordered region. And we can also do this with MSAs. So here's same thing again with an endosome, lysosome, vesicle-associated protein. And here, this is slightly more similar. It's 37% similar between the generated and original sequences. So you can see we do a decent job of keeping the disorder predictions where we should and the order predictions where we should. Of course, you might wonder, can we just do this with a random model or something else that's much simpler? And the answer is not really. So if we look at the distribution over a larger sample of things we generated, first in gray are the true predictions, and those are the same in each block. Or they're different in each block because we use different IDRs for EvoDiff-seq and EvoDiff MSA. And in the disorder regions, those are obviously going to be higher than they are in the structured regions. Then in blue, we show predictions when we regenerate the IDRs with EvoDiff, either seq or MSA. And so you can see we're not as separated as the natural ones, but we are doing much better than the random ones in red. And then if we look at sequence similarity, as you would expect, because the EvoDiff MSA has so much more information, it does tend to generate more similar IDRs to the native than EvoDiff-seq. All right, so we've talked about regenerating queries. We've talked about trying to purposely generate disordered regions. Next, we're going to talk about motif scaffolding. And the motivation here is that many protein functions are mediated by what people call a motif, which is shown in purple here, stabilized by a scaffold shown in gray. And these things include epitope presentation, our immune system, viral receptor traps, enzyme active sites, and protein-protein interactions. I stole this picture from a science paper from Baker Lab, but the general intuition here is that there are some protein engineering tasks or protein design tasks where you can specify an active site or binding site in 3D space, and then you just want to scaffold that, a scaffold that holds that in place, so such that you can do the function. And people have come up with a variety of ways to use structure predictors and then use structural diffusion models to do this. And we wondered, could we actually just do this directly in sequence space if we knew where the motif was that we wanted? And it turns out that, of course, I wouldn't be talking about this if you couldn't. So here is PDB1PRW, which is binding site of compact calmodulin. In green, we've shown the binding motif. In gray is a scaffold. And we show the whole sequence below. And here, I'm going to just shift where the motif is in the sequence, and then mask out the rest of the sequence and regenerate the scaffold. And for some reason, I decided to do this with the actual amino acids we generated and the actual native ones and the actual positions, which meant this took a long time to do. And you can see that the omega fold predicted structure, which holds that active site, that motif, in place in pretty much the same conformation. We do this without needing structure at generation time. All right, so let's look at some more examples of this. So here's an example that we did with EvoDiff seek. This is 1PRW. And this is actually the same one we showed in animation. You can see the native structure. We take out the functional motif. Sorry, this is actually a different one from the one we showed in the animation, but also successful. And then we look at the predicted structure after generation. We see it has high confidence, low RMSD, less than 1 angstrom on the functional motif, and a fairly low TM score, 0.5, to the original native structure, even though the motif itself is very conserved. We can also do this with EvoDiff MSA. This is a 5YUI. It's a binding site of the carbonic anhydrase metalloenzyme. And so the native, this is a pretty complicated one. The native structures on the left, this has a, well, both of these actually have disjoint functional motifs in sequence space. We generate a much smaller scaffold, but it has very high omega fold PLDT. The motif RMSD is less than half an angstrom, and the TM score is 0.88. So even though it's much smaller, the parts that are preserved are actually pretty similar structurally to the original. So we looked at a set of 17 problems that were first compiled in the RF diffusion paper. And RF diffusion, remember, is a state-of-the-art structure diffusion model. And for each of those 17 problems, we sampled 100 scaffolds using RF diffusion, EvoDiff MSA, and EvoDiff seek. So you can see in the second column, we consider a scaffold to be successful if the motif has less than 1 angstrom RMSD to the desired motif. And so giving you structure does help. In general, you do have a higher success rate, but the success rate is significantly non-zero using MSAs or sequences alone. And then the rightmost column is out of the 17, how many have you come up with at least one solution for? You see that RF diffusion, EvoDiff MSA actually have solve the same number of problems. EvoDiff seek solves slightly fewer because it gets less information. All right, so we can also just compare how well they do in each of these problems. So here I'm showing the number of successes per problem, and the problems are named by their PDB file, their PDB structure. So there's 100 attempts per problem, and we're comparing EvoDiff seek versus EvoDiff MSA. You can see that on almost everything, where any of them have a success. For almost everything, EvoDiff MSA has a higher success rate. Although that's not always the case, somewhat surprising. We can also compare using EvoDiff MSA versus RF diffusion. And the interesting thing to me here is that they have pretty orthogonal strengths. There's not a whole lot of things where both have a high success rate. 1 BCF and 6E, 6R, and maybe 6EXC. But otherwise, they tend to do well when the other one does not. Some other things that stick out, 1PRW, which is an example in animation, is the one weird one where EvoDiff Seek does better than either EvoDiff MSA or RF diffusion. So 3IXT is one where both EvoDiff Seek and RF diffusion do better than the MSA model. Looked at that, and we actually had trouble finding homologs for this one, which makes sense because your ESM model, your MSA model is dependent on having good homologs. And then we also looked at how similar are the successful scaffolds to the native scaffolds. And here I'm plotting for every problem where both have at least one successful scaffold, the TM score between the generated and the original. And for almost all of these, the one generated by EvoDiff Seek is more different from the original. And that makes sense because you're not giving it that implicit structural information from the MSA.
Hey, Kevin, just a reminder, you have about
Yes.
10 minutes left.
Yep, we are good. So then we talked about EvoDiff, which is an evolutionary scale discrete diffusion model for controllable protein sequence diffusion. And the vision here is that we want to learn from natural sequence space, and we want to focus on sequence space because it's universal design space for proteins, and there's just so many more of them than we have structures. And we know that natural sequences encode some structure, which for a lot of things is a nuisance variable that we want to integrate out. And that does something in the cell. We train EvoDiff on things from across evolution and across function. And then hopefully we can use EvoDiff to generate new sequences that do new desired functions. We covered that EvoDiff can give you high quality, unconditional generation. We can do conditional generation with EvoDiff, including this motif scaffolding completely and directly in sequence space without explicit structures. And then looking ahead, we want to do controllable generation. So we'd like to specify the desired function eventually and generate sequences directly to do those functions. So that's where me and my colleagues will be focusing on for the future. If you would like to try these models, they are open source. You can see the training code, you can see the data sets, you can see the weights. There are scripts to load and use models. And the best place to start is the GitHub. And with that, I would also like to thank some of these people are authors in this paper, but everybody else is in the BioML group at MSR New England, which is just a great place to work and to do this kind of open, interesting research at the intersection of machine learning and biology. With that, I will take questions. Well, there are 43 things in the chat box. Okay.
Awesome. All right, you want me to read some of the questions from the chat, Kevin?
Sure, if you want to triage them a little bit.
Yeah, sure. So skipping to one of the questions here, what do you think about the impact of synonymous codons on folding behavior? Benjamin asks, particularly around the idea that different codons have different functions in cells pertaining to like the rate of binding and protein synthesis.
Right. So we're kind of taking a simplified modeling perspective here where we only consider the amino acids and we actually synthesizing and translating these is a problem for later. But I agree. The actual codons you use matters making these, and it depends on the organism and different mass language models and maybe in autoregressive language models on the codons instead of on amino acids, which I think is really interesting. But we did not do that. So that is a problem for later us when we decide we want to make these in the lab.
All right. Simon asks, if the D3PM model didn't train to convergence, do we really know that OADM performed better?
We do train them for comparable amounts of time before we decide which one to go with. The OADM model is definitely not trained in convergence. We could probably train it longer. That involves asking for more GPU time, which is always a fun thing to do with the lab. Yes, but we do compare at the same number of training steps.
All right, combining two questions that are sort of related here. Jeff asks, am I misinterpreting? The Hamming distances are quite small, suggesting they're quite similar, albeit not exact to things seen in training. And then there's a similar question down below that basically asked, when you're doing kind of the conditional generation with IDRs, are you like concerned at all about kind of just reconstructing the parent sequence?
Right, so with the IDRs, let me find that. This is a slow part, because it's much complex. Okay, so, sorry, I'm kind of lost in my own presentation now. Here we go. Next slide. So if we look at these, so probably this is something to be more consistent with, because sometimes I show Hamming distance, and sometimes I show Hamming similarity. If you look at these are actually similarities. But those are fairly low, right? Those aren't like 90 or 80 even. And if we look at the distribution of similarities, there are definitely, EvoDiff pretty much doesn't generate anything over 50. EvoDiff MSA in some cases does reproduce almost the exact same thing, but we definitely did still manage to pick out cases where they're pretty different and still preserve that expected predicted disorder. I should probably more consistently stick with either similarity or Hamming distance, because they're inverses of each other.
And then a sort of related question, Benjamin asks, if the sequences are all the same length, wouldn't the Levenstein distance be a better metric than Hamming, closer to what happens in evolution?
Yeah, we are trying to go back and compute either Levenstein distance or like blast e-values for a lot of these, because we agree those are better metrics. And we thought of this kind of late in the process, and those computations are not that fast when you've generated thousands of things.
Great. Just scanning through a couple more questions here. Rakesh asks, is it fair to compare both sequence and structure-based diffusion models like RF diffusion using the FPD metric? The reason you'll find lesser sequence diversity in UMAP-based embeddings for structure-based models is that only a subset of sequences can form a structural scaffold.
Right. It is definitely unfair, and the argument we're making there is that going into it, you know it's going to be unfair, right? Because you're training a structure-based model only on structured domains, and not very many of them. But in science, it's always good to go and confirm that what you think is true is actually going to be true. And the takeaway there is that if you want to generate across the full breadth of possible proteins, training a sequence might give you some benefits.
All right, I think there's maybe one or two more questions after this one. Avik asks, the plot model that you used is more rudimentary. There have been better versions of the model that have been shown to perform better than neural nets for capturing epistasis. One example is McGee Nature Communications 2022. Do you think that these POTS models may actually perform on par with some of the diffusion models here?
Right, so I would not actually be that surprised if you curate a high quality MSA and you spend some time tuning your POTS model, you could get very similar and possibly even better performance than any neural network model trained into the side MSA or on all MSAs. And the reason I say that is because when it comes to variant prediction, we still see that these fairly simple MSA-based methods are still very, very, very strong.
All right, I think we have maybe two more questions here. And I don't know if you have time to stick around after 5, Kevin, but certainly we can stay on until a few more minutes at least.
I see Sarah's in the chat answering questions for me. Thanks, Sarah.
Oliver asks, can this be used as sort of an affinity maturation tool? Could I guide EvoDiffusion to suggest mutants likely to bind better to my target?
Yeah, so Brian He did a very similar thing with ESM, right, which is a mass language model. And the hypothesis there was that you just use it to filter out the mutations that are going to break your antibody. And then that allows you to only screen things that will produce valid antibodies. And then that raises the probability of finding valid things that are strong binders. And I suspect you would be able to do the same thing with this sort of model.
All right, last question here. Gurvinder asks about your next steps of controllable generation for a given function in sequence space. Do you have any ideas which data sets can be relevant?
We have several ideas, which, because of agreements I've made with other collaborators, I cannot necessarily share in a public seminar. But we definitely have a lot of ideas, and if you're interested, one-on-one, I might be more forthcoming about those.
Awesome. Oh, and then I also meant to ask, a lot of people in the chat have been asking about experimental verification. Oh, and I actually just messaged in the chat right as I said that. Are there plans to kind of spearhead efforts to validate these, new proteins in the wet lab, or will that kind of be something that other groups explore with the model?
We are working on that ourselves. If you have ideas for things you would like to try with this in the wet lab and like to talk to us, we would also welcome that. But yes, we are definitely doing that.
All right, wonderful. Well, if there are no other questions, let's thank our speaker for talking about some of this exciting new work out of his group. And thanks to everyone for joining us. If you haven't already, don't forget to subscribe to our channel on Twitter. We have a Slack link as well where we talk about developments and protein engineering and maybe some job postings and networking, things like that. You can also find the rest of our seminar schedule on our website, where we've posted, I think, talks until the end of October, and more speakers to be planned for the rest of the fall after that. All right, so thanks again for everyone who are coming. Have a great afternoon.
All right, thanks so much. Really great being here.